Hello and welcome or welcome back to NEET Prep Go. So today we'll be doing another chapter, Animal Kingdom. Again, it will be a fast revision of the chapter Animal Kingdom. So when you look around, you will mostly first we'll do non-chordates and then we'll do chordates. First, let's get started. When you look around, you will observe different animals with different structures and forms. As over 1,000,000 species of animals have been described till now, the need for classification becomes all the more important. Classification also helps in assigning a systematic position to newly described species. So what does classification also help? That is, classification also helps in assigning a systematic position to a newly described species. Now, basis of classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory and reproductive system. These features are used as the basis of animal classification and some of them are discussed here. Firstly, level of organization. So in spite of so many differences in structure and form of different animals, we need to classify, right? To assign a systematic position to a newly described species. So therefore, there are fundamental features on which various individuals can be classified, that is arrangement of cells, then body symmetry, whether they are asymmetric, bilaterally symmetric or radially symmetric, nature of coelom, whether they are pseudocoelomate, acoelomate or true coelom, that is they possess a true coelom, then patterns of digestive system, open or closed, circulatory system, open circulatory system, that is no blood vessels or anything, closed circulatory system, blood flows in veins and capillaries. Digestive system is not open and closed, it is complete incomplete, that is complete digestive system having both openings, mouth and anus, whereas incomplete having only one opening. Then reproductive system and reproductive systems. These features are used as the basis of animal classification and some of them are discussed here. Firstly, levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern. So we know that bacteria are unicellular prokaryotes, then your kingdom Monera, then kingdom Protista are unicellular eukaryotes, then kingdom Fungi are multicellular eukaryotic organisms, then after that, that is your Plantae and Animalia, all of them are multicellular and all of them are eukaryotes, but all of them do not exhibit the same pattern of organization of cells. That is, they are not completely cellular, but they can be cellular as well as, that is, organ system, that is they exhibit lot of levels of organization. For example, in sponges the cells are arranged in loose cell aggregate. In sponges, that is porifera, in which sponges come, that is the cells are arranged as loose cell aggregates. They exhibit cellular level of organization. Some division of labor, that is activities, occur among the cells. In coelentrates, the arrangement of cells is more complex, that is they arrange the cells performing the same function are arranged into tissues. So porifera has cellular level of organization, whereas coelentrata or Nidaria has tissue level of organization. This is tissue level of organization. Then all the tissues, they together combine to form an organ. So still higher level of organization is the organ level of organization, which is exhibited by members of platyhelminthes and other phyla, where tissues are grouped together to form organs, each specialized for a particular function. In animals like after platyhelminthes, that is from aschelminthes till your hemichordata, all of them exhibit organ system level of organization. Functional system, each system concerned with a specific physiological function, this is called as organ system level of organization. Organ systems in different groups of animal exhibit various patterns of complexity. The digestive system in platyhelminthes has only a single opening to the outside of the body that serves as both mouth and anus, so therefore it is incomplete digestive system is present in platyhelminthes. Complete digestive system has two openings, mouth and anus. Circulatory system can also be of two types. So till platyhelminthes there is incomplete digestive system. After platyhelminthes, that is from aschelminthes to your hemichordata, that is all have complete digestive system. So from aschelminthes to hemichordata, that is complete organ or that is organ system level of organization, as well as your complete digestive system. Then a complete digestive system has two openings, mouth and anus, whereas incomplete digestive system only has one opening, which serves as mouth as well as anus. Similarly, the circulatory system can be of two types, that is open type and closed type. Open type circulatory system, in which blood is pumped out of the heart and the cells and tissues all are directly bathed in it. For example, in arthropoda, then echinodermata, sorry, in echinodermata the water, in arthropoda, hemichordata, then non-cephalopod mollusks, and yeah, that much only. In Annelida it is closed, in mollusca also it is closed. Till aschelminthes there is circulatory system is not present only. Closed type of circulatory system, in which blood is circulated through series of your vessels, arteries, that is arteries, veins, capillaries of varying diameters. Smallest being capillaries and largest being arteries. Radial symmetry. Okay. So then we come to symmetry. That is, if we take a sponge, okay. So if we cut it like this, is it in any plane, whether it be like this, whether it be like this, whether it be like this, are we dividing it into two equal parts? No. So this is asymmetrical. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical. Any plane that passes through the center does not divide them into equal parts. No matter how many planes in whatever plane you pass through, the two halves are never going to be equal. So therefore, they are asymmetrical in nature. When any plane passing through the central axis, so your poriferans are asymmetrical in nature. Then, when any plane passing through the central axis of the body divides the organism into two identical halves, that is in any plane. So if I take, this is not a good diagram. So if I take uh, an organism, how many planes I no matter how many planes I pass through, I'm always going to get identical. From the center, I'm going to get identical. From here also, I'll get identical. So then we call it radial symmetry. It can be seen in Coelenterata or Cnidaria. Then it can be seen in this. This I was actually trying to draw Tinoplana, failed miserably. So in Tinophora, that is Tinophores, and in Echinodermata, your starfish. It is all, they are all radially symmetrical. No matter how many planes you pass, that is any plane you pass through, it is, you are always going to get two equal halves in any plane is called as radial symmetry. Then bilateral symmetry, in one place, sorry, in one plane, if you, that is only in one plane you get two identical halves, then it is called as bilateral symmetry. That is animals like Annelida, Arthropoda, um, mollusca, aschelminthes, platyhelminthes, that is from that is your from so first porifera is going to be asymmetrical, then your Ctenophora and Cnidaria or Coelenterata is going to be radially symmetrical, then platyhelminthes, aschelminthes, Annelida, Arthropoda, mollusca, they are all going to be bilaterally symmetrical. Then Echinodermata are going to that is adult echinodermata are going to be radially symmetrical, whereas baby, that is juvenile before adult reaching stage, that is your Juvenile echinoderms which are not yet adult, pre-adult stage, they are going to be bilaterally symmetrical, and hemichordata is also bilaterally symmetrical. Here adult echinoderms. Then where the body can be divided into identical left and right halves in only one plane, they exhibit bilateral symmetry. Now diploblastic and triploblastic organisms, animals which have an ectoderm and an endoderm, they are called as only two embryonic layers, external ectoderm and internal endoderm. They are called as diploblastic animals. Example is coelenterata, coelentrates. An undifferentiated layer, mesoglia, is present between the endoderm and ectoderm in diploblastic organism, that is in coelenterata and cnidarias. Sorry, coelenterata, cnidaria, same thing in ctenophora. Diploblastic organism. Then animals in which the developing embryo has a third germinal layer, that is mesoderm, in between the ectoderm and endoderm. They are called as triploblastic animals. Platyhelminthes, still chordata, all of them are triploblastic animals. Then coelom. What is coelom? Coelom means a cavity. So presence or absence of a cavity between the body wall and the gut wall is very important in classification. Body cavity which is lined by mesoderm is called as coelom. The body cavity which is lined by mesoderm is called as coelom. So these are acoelomates. Let us see, there is no cavity, so acoelomate. So till your porifera till platyhelminthes, all of them are acoelomates. Your aschelminthes alone is a pseudocoelomate. This is pseudocoelomate, wherein the coelom is present in that is patches. So animals possessing coelom are called as, and after aschelminthes, that is from Annelida till your chordata, all of them are coelomates. So your Annelida, mollusca, arthropoda, echinodermata, hemichordata and chordata, all of them are coelomates, that is they possess a true coelom. They are also called as eucoelomate. Then some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present in scattered pouches in between the ectoderm and endoderm. That is, the mesoderm is present in scattered pouches. So there is cavity, but mesoderm is present in pouches. So this is called as pseudocoelom, and possessing them are pseudocoelomate. Example is aschelminthes. Then animals in which body cavity is absent completely are acoelomates. Example, your porifera till platyhelminthes. Then segmentation. In some animals, the body is externally and internally divided into segments with serial repetition of at least some organ. For example, in earthworm, the body shows the pattern called metameric segmentation. This phenomenon is called as metamerism. So what is segmentation? That is when the body is externally and internally divided into segments with serial repetition of organs. That is heart, 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 heart. That is serially, they will keep getting repeated. That is in segments it is divided, and some organs will be repeated in each and every segment. This is called as metameric segmentation, and it is found in earthworm. That is, true segmentation is found in Annelida, and this phenomenon is known as metamerism. In arthropoda, only external segmentation is found, and in chordates, I think only internal segmentation is found. I'll have to check once. Notochord we'll see after this. Notochord is mesodermally derived rod-like structure. What is notochord? Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called as chordates, and those which do not form this structure are called non-chordates. So this notochord is mesodermally derived firstly, and it is a rod-like structure, and it is formed in the dorsal side. You'll see in cockroach and all, there is a ventral nerve cord. It is not a notochord, it is a ventral nerve cord is there. Notochord, that is till your hemichordata, or they are all called as non-chordates. That is, okay, sorry, porifera to echinodermata, they are all non-chordates. They do not possess a notochord. I think till hemichordata you can say. That is, the notochord type structure is rudimentary in hemichordata. Anyways, Classification of animals. Broad classification of animalia based on fundamental features mentioned in preceding section, figure 4.4. So, kingdom, animalia, multicellular, eukaryotic, levels of organization, that is cellular level of organization is porifera, then tissue level of organization is Coelenterata and Ctenophora, organ level of organization is Platyhelminthes, organ system level of organization is Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. Then radially symmetrical is Coelenterata or Cnidaria and Ctenophora and adult Echinodermata. Then bilaterally symmetrical is your Platyhelminthes, and porifera is asymmetrical. Then bilaterally symmetrical is Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca. That is not adult. That is pre-adult, juvenile, before the adult stage. Bilaterally symmetrical echinoderms are there. Then hemichordata and chordata. See, here it is written echinodermata exhibits radial or bilateral symmetry depending on the stage. In adult stage, it is radially symmetrical in adult stage. Before that, it is bilaterally symmetrical. Then without body cavity, acoelomates are porifera, coelenterata or cnidaria, ctenophora and platyhelminthes. With false coelom, pseudocoelomate is only one, aschelminthes, and true coelom, that is or eucoelomates, true coelomates or eucoelomates. They are Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. The important features of different phyla are described below. So let's see this once again very fast. So porifera, coelenterata or coelenterata or cnidaria, then ctenophora, platyhelminthes, aschelminthes, annelida, arthropoda, mollusca, echinodermata, hemichordata. Porifera has cellular level of porifera has cellular level of organization. Then your coelenterata as well as ctenophora, both of them have tissue level of organization. Platyhelminthes alone has organ level of organization, and from aschelminthes till hemichordata, all of them have organ system level of organization. Then comes your digestive system, that is in porifera there is no digestive system is present because it has only cells, that is in the loose cell aggregates are present in porifera. In coelenterata or cnidaria, that is incomplete digestive system is present. In ctenophora also incomplete digestive system is present. In platyhelminthes also incomplete digestive system is present. From aschelminthes complete digestive system is present till hemichordata. Then comes your circulatory system, that is in porifera, coelenterata, cnidaria, ctenophora, platyhelminthes, that is digestive system is not circulatory system in aschelminthes also till aschelminthes digestive system is not present. Only till aschelminthes digestive system is not why am I saying digestive system, circulatory system. Digestive system तो इसमें porifera में नहीं था. Then coelenterata और cnidaria में there is incomplete digestive system. Ctenophora incomplete digestive system. Platyhelminthes incomplete digestive system. That is only one opening for both mouth and anus. From aschelminthes till hemichordata, complete digestive system is there. Then circulatory system not present till aschelminthes. So circulatory system very important. Circulatory system not present till aschelminthes. Till aschelminthes circulatory system is not present. After aschelminthes, that is In Annelida, then uh, cephalopod mollusca. In Annelida and cephalopod mollusca, closed circulatory system is present. That is, blood flows in arteries, veins and capillaries, whereas in arthropoda, non-cephalopod mollusca, that is those that do not contain shell. Then echinodermata. In echinodermata, actually there is a water vascular system, so again no circulatory system. Hemichordata, open circulatory system is present. That is, blood directly from the heart pumps the blood, and it is directly bathed in organs and tissue. That is, the cells and tissues are directly bathed into it. Then your body symmetry. That is, in porifera it is asymmetrical. In Nidaria or Coelenterata, Ctenophora, and Echinodermata, adult Echinodermata, there is radial symmetry. And in your Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, and juvenile or pre-adult Echinodermata and Hemichordata, it is bilaterally symmetrical. Bilateral symmetry. That is, only in one plane you can divide. Then germ layers, that is, this has no germ layers, only loose cell aggregate. In your Ctenophora, Nidaria or Coelenterata and Ctenophora, there is they are diploblastic. And your rest, that is from Platyhelminthes till your Chordata, all of them are triploblastic. That is, they have three germ layers: ectoderm, endoderm and mesoderm. Whereas they are diploblastic, they have ectoderm, endoderm. In the middle of ectoderm and endoderm, they also have mesoglia, which is undifferentiated. Then that is, jelly-like undifferentiated layer, mesoglia is also present. Then coelom or body cavity, that is porifera, from porifera till Platyhelminthes, all of them are acoelomate. Then Aschelminthes is pseudocoelomate, and from Annelida till Hemichordata, all of them are eucoelomate or coelomates. Out of those also, Annelida, Arthropoda and Mollusca are schizocoelomate, whereas Echinodermata and Hemichordata are enterocoelomate. That is your Your Schizocoelomate, that is, schizocoelomates are developed by splitting up of mesoderm, and they are found in Annelida, Arthropoda, and Mollusca. So in your what happens? In your Annelida, Arthropoda, and Mollusca are schizocoelomates. Why? Because they are made up of splitting up of mesoderm. Splitting up of they are called as schizocoelomates because they are made up of splitting up of mesoderm, whereas your echinodermata and hemichordata, they are called as enterocoelomate. Why are they called as enterocoelomate? Because they are made up of embryonic gut or enteron, from the wall of embryonic gut or enteron, as hollow outgrowth or enterocoelomic pouches. They are made from wall of embryonic gut or enteron. Then segmentation, that is, annelida is externally and internally segmented, so it shows true segmentation, that is internal plus externally segmented. Arthropoda shows only external segmentation. Arthropoda shows only external segmentation. Okay. So now let's get back to the chapter Porifera. Members of this phylum Porifera are commonly known as sponges. They are generally marine and mostly asymmetrical animals. They are primitive multicellular animals and they have cellular level of organization. Sponges have a members of phylum Porifera are commonly known as sponges. They are generally marine and mostly asymmetrical animals. They are primitive multicellular animals and they have cellular level of organization. Primitive multicellular animals, cellular level of organization, that is loose cell aggregates. Sponges have water transport system, also called as canal system. Water enters through minute pores or ostia in the body wall into a central cavity which is called as spongiocele. From the central cavity or spongiocele, it goes through the osculum. The pathway of water transport is helpful in food gathering, respiratory exchange, and removal of waste. Choanocytes are collar cells which line the spongiocele or the central cavity and the canals. Digestion is intracellular. Body is supported by a skeleton made up of spicules or spongin fibers. Sexes are not separate, that is hermaphrodite. Eggs and sperms are produced by the same individual. Sponges reproduce asexually by fragmentation. Sponges reproduce asexually by fragmentation or sexually by formation of gametes. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. Examples is cycon, also called as cypha, then spongilla, which is a freshwater sponge, and euspongia, which is bath sponge. Spongilla is a freshwater sponge, rest both are marine. Cycon or cypha, euspongia. So this is cycon, also called as cypha. This is uh, euspongia, freshwater sponge. This is spongilla, bath sponge. So they are pore bearers. They are all marine except for Spongilla, which is a freshwater sponge. They have a water canal system or water transport system. So in phylum Poriferans, they are commonly called as sponges or pore bearers. They are generally marine, mostly asymmetrical. They are generally marine except for Spongilla, which is a freshwater, use. Except for Euspongia, which is a Spongilla, not Euspongia, Spongilla, which is a freshwater sponge. Euspongia is bath sponge. They are primitive, multicellular, they have cellular level of organization, they have a water transport or canal system. Water enters through minute pores or ostia in the body wall to a central cavity called as spongiocele, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, removal of waste. Choanocytes or collar cells, they line the spongiocele and the canals, and as well as the canals. Then a body supported by skeleton, spicules or spongial fibers. Sexes are not separate, hermaphrodite. Fertilization is internal. Digestion is intracellular. Sponges reproduce asexually by fragmentation, sexually by formation of gametes. Fertilization development indirect. It has a larval stage which is morphologically distinct from the adult. Then phylum Coelenterata or Cnidaria. Phylum Coelenterata or Cnidaria are aquatic, mostly marine. They are aquatic, mostly marine. They can be sessile, that is restricted to one place, or free swimming. They are radially symmetrical animals. The name Cnidaria is derived from nidoblast or nidocytes, which contain stinging capsules, or nematocytes present on the tentacles and the body. Nidoblasts are used for anchorage, defense, for capture of prey. Cnidarians exhibit tissue level of organization. They are diploblastic. They have central gastrovascular cavity with a single opening, mouth on hypostome. Digestion is extracellular and intracellular. Some of the cnidarians corals have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body, polyp and medusa, like Hydra, Adamsia, whereas we'll do this part in the next day.